Hello dear student, good morning. So this is the second lecture of the cell and tissue culture technology. In the previous lecture we have learned about the scope of the cell culture, some basic definitions, some glimpse of the history. So repeating the first lecture, just like the glimpse of the first lecture, that the tissue cell and the tissue culture was a technique practiced about 100 years ago, and that was the Harrison in 1907 who observed the development and growth of the nerve fibers, and then in 1912 the attempt was made for the state-of-art cell culture experiment, and the chick embryo tissue was grown in that experiment. So this was the little bit history of the cell culture and technology. So the cell culture and technology, I previously told you that these are the advantages of the cell culture and technology, that we can control the surrounding environment, and then it is very simple and homogeneous technique, and we can use the specific cells, and it is very cheaper whenever we plan some research experiment, and we can practice at very economical cost. The experiments we can conduct at very economical cost, and then we can get the results in very short time. And the next is the why we go for the cell culture experiment, that this is every time possible. We can create the anytime we can create the environment that we can practice the cell culture technology at every time. So in this lecture we will learn some more concept of the cell culture and technology. So here is the slide about the cell line. What is the cell lines? The cell lines are the culture of the animal cells that can be propagated repeatedly and sometime indefinitely. They arise from the primary cell culture. Now the question is that what is the primary cell culture? Primary cell culture can be initiated directly from the cells, tissue and the organs of the animal, which are typically used in the biomedical research those days. So if we define the cell culture, primary cell culture in more detail, the different type of culture they are nowadays routinely performed, and these cultures can be divided as the basic type, that is the primary cell culture. So primary cell culture can consist of the culture of a complex organ or tissue, slice a defined mixture of cells. We can say that a mixture of cells or highly purified cells which can be directly isolated from the organs of the body, and this is you can say that more commonly used techniques nowadays used in the cell culture lab. So these primary cell culture, usually because the primary cell culture is isolated directly from the multicellular organs, so whenever we isolate these from the organs, there are certain advantages and certain disadvantages. So we have two choices: the primary cell culture and the established cell lines. These are the two choices we have whenever we do the cell culture research. So there are some advantages of primary cell culture, that they having a very good presentation, and they having the heterogeneity, but they having some disadvantages over the establishment cell line. This is the slide about the cell line. I will tell at the end that of this slide that what is the disadvantages of the primary cell culture over this established cell line. This cell line please first learn about the established cell line. What is the cell line? The cell line is permanently established cell culture that will proliferate indefinitely given appropriate fresh medium and space. A cell culture developed from single cell and therefore consisting of cells with a uniform genetic makeup. So I previously told you that the primary cell culture having this advantage here, that primary cell culture having a very good level of heterogeneity, and primary cell culture having, I previously told you that there is an advantage, but there is some the disadvantages of primary cell culture over the established cell line. So what is the disadvantages of primary cell culture over the established cell line? The first advantage, which is a very big advantage, that is the damage to the sustainable damage or partial damage to this cell line, to this primary cell culture whenever we isolate them from multicellular organisms. So the other the cell line, this is another slide. This is the continuation of the well established cell line. So subculturing of primary cells that lead to the generation of these cell lines. Whenever we subculture the one type of cells, or we isolate the certain type of cells and develop, that is the called the established cell line. So what is the continuous cell line? The most continuous cell line grow for the limited number of the generation, after which they cease. We grow them for a specific period of time and then stop growing them and stop. So here these continuous cell line may be ceased. The cell line which either occur spontaneously or induced virally are chemically transformed into the continuous cell lines. So this is about the continuous cell line. So talking about the cell lines, the established cell line having some more advantages in the recent area of the research. The primary cell line are the first type of the cell cultures we perform, and then the well established cell lines, this is the second type of the cell culture. The vast majority of these cell lines basically these are derived from the tumors and from the cells or from the other cells which are transformed in vitro. So although there were very earliest, there were some cell lines which were established from the embryonic tissue, but there are some other cell lines that have been widely used, like an example of WI38, usually which we use the normal human tissue and have a very limited lifespan whenever we practice them the in vitro research. These cell lines are nowadays, I am using the word, the workhorses of the cell culture nowadays. So they from their use in study to control of the cell culture to vaccine production, or if we study the large scale vaccine production. So these cell lines are not surprisingly from many decades in the laboratory, they are both relatively they are the scientists are using, and the scientists are using so repeatedly that by culturing techniques their original phenotype is changed, and these cells having the the same designation they have carried in the different laboratory, and maybe they are varying in the properties because of the variant changes in the phenotype. Maybe they having some changes in the properties, but commonly we use these cell lines for the different exercises we do, like for the vaccine production, and some some immunity assays, or some drug testing, or some enzyme testing, or peptide experiment, protein experiment, or cryopreservation conservation studies, we every time go for these established cell lines. So this is another slide telling you about the types of the tissue culture. You can see the primary cell culture, and then the continuously cell culture, and then the continuous cell culture having the two type. The finite single cell type, roughly 30 time of the division enhanced by the growth factors, and then the indefinite, it is nearly the same as the finite, but the cells here can divide indefinitely by transformation into the tumor cells. They are called the cell line. So more recently, a cell line have been developed with the aim of maintaining their normal phenotype, combined with the ability to grow the cells or its precursor indefinitely in the culture. So this is another slide which can tell you about the three type of cell line. The cell line, this main type is the continuous cell line, that when a finite cell line undergoes transformation and acquires the ability to divide indefinitely. Then the finite cell line, the normal cells usually divided only a limited number of time before losing their ability to proliferate, which is genetically determined event, known sentences. And then the transformed cell lines, so conversion of normal healthy cell into the tumorous and cancerous cells. So these are basically three types of cell line we usually practice in day-to-day cell culture and technology research. These cells usually depend on the requirement of the research work, that which type of research work we are doing. If there is an example of wound healing studies. If whenever we do the wound healing studies research, we don't use the transformed cell line, so we go for the normal cells usually. So finite cell line we use. Like another the skin graft studies also, they usually using the skin cells, and they go for the finite cell line. Also for the peptide studies, the cytotoxicity studies also depend on the type of studies here. Now here is the game for the research, that what type of research you are doing, what type of the research journey you are being carrying out. So sometimes you have to the different type of choices. If your research is related to the cancer research, then you will definitely go for the transformed cell line. If you have some research where you need the continuous experiment of growing and growing and growing again, like for the vaccine studies or some type like this, so we go for the continuous cell line. And sometimes we have a very short experiment, and we just want to do some cytotoxicity assays or some cell viability assays, or like some drug testing or enzyme testing or purification studies like this. So we go for the finite cell lines. Basically, the aim of the mammalian cell culture is to provide an environment that mimics the great extent possible. In vitro environment of that specific cell type, we go for different type of experiments, but these all experiments depend upon that the environment we create in the cell culture laboratory. The cell culture, I'm just giving you an day-to-day example when usually we practice in the cell culture lab, that the cell culture incubator, the culture dishes, the apparatus or the medium you are using, these all together they create an environment for in vitro research. They are very helpful in providing an appropriate temperature. I am talking about the pH, the oxygen, the carbon dioxide supply. These all steps, the surface for the cell attachment, the nutrients, the vitamin supply, the protection against the toxic agents, the hormones and the growth factors. These all we can say that control the cell states and growth of differentiation. These all things depend usually when we are doing the cell culture experiments. Here is another slide about the finite type of cell lines. These are the name of the cell line. We have 4 columns in this slide. So the cell line names are IMR 90. These are very commonly using cell lines. Some we use for the research in the cell culture labs, and they are originated basically for lungs. Species, we use the human species of lungs, and they are very susceptible for the human viral experiments, infections. So whenever we do some experiments like where we need the susceptible cells to the human viral infection, we use this type of cell line. Then another MRC5, these are also isolated from the lung tissue, human lung tissue, and these are also very susceptible to the human viral experiment. The next two are the this 293, that is from the human kidney. So this is another slide, continuous cell line, CaCO2. Then a friend, which are the CaCO2 is originated from colon, friend is originated from the spleen, but spleen of mouse, here from the lab animal. So characteristic that they having hemoglobin characteristic, and then P388D1, these are also mouse spleen oriented cells. So they can grow into the suspensions. Then GH1 and GH2, these are rat cells, originally basically from the spleen isolated rat cells. Growth hormones, they are very reactive to the growth hormones. So whenever we do some studies that involving the growth hormone, we prefer we can prefer this type of cell line. Also S180, these are from the mouse oriented cell line. So this is a cartoonic representation about the cell culture. You can see that how we perform the cell culture. You can see that the isolated cell culture, and then the growth of the cell culture, and then the dissociated 2D culture. So this organ can this presentation, you can see this cartoonic presentation can tell you something about the some certain type of culture techniques. So the early scientists of the cell culture, they used exceedingly complex environment to maintain their pieces of tissue culture or cell culture when they were growing it in vitro. They including the plasma clot and the tissue extract in a complex hand blown glass chambers. They designed to maintain the sterility while providing the gas exchange. Then with the use of the better and more complex media, then coming the serum supplements into the scene, alone was sufficient basically to grow many transformed cell types in the cell culture, although the cells still required the some more supplementation. But this was usually the 1950s, where we were able to feed the cells in vitro techniques. So our talk is continue that how this cell culture technology become more possible and more suitable technique it's become in the recent area. So this was another this is the fetal bovine serum, you can say, and the fetal bovine serum, this nowadays very commonly we use in the cell culture. Like you can say that this is a mainly used chemical in the cell culture research. So what is the importance of the fetal bovine serum? Fetal bovine serum, they compose of the growth factor and the hormones. What they do, they add the cell attachment, they binds the toxins, they neutralize the toxins. So this is also it have very a long history of use in the cell culture laboratories. Um, they having very different compositions also. It is an expensive item of cell culture research whenever we do cell culture experimentation, and we use it very regularly to minimize the growth and to minimize the risk factors. And there we can, this is the heat inactivation. If because of the destruction of the complement and immunoglobulins at this temperature, if we keep it at 56 degrees centigrade for 30 minutes. And also so this was something about the fetal bovine serum. Fetal bovine serum is a very basic component nowadays in the cell and tissue culture research. Because of the advantages of there are many advantages of fetal bovine serum, we always use and we cannot skip some substances of the cell culture research. There is another slide. This is the slide telling you about the culture media, that a growth medium or culture medium, it is a liquid or gel that is designed to support the growth of microorganisms or the cells. The components of the suitable cell culture media include the basic media, the buffering capacity, the glutamine and other amino acids, also the serum, then the antibiotics and the antimycotics, and then the choice of the culture media used that depend on the type of the cell culture, that we are using the primary cell culture, or we are using the cell lines, or we are using the definite these other type of cells we are using. So and these are the basic three type of media here. It is you can see in this slide the basal media. There is another type which is called the reduced serum media, and then the third type is the serum free media. So there are usually these three type of media we usually use in cell culture technology and cell culture research. Today lecture is very important if you want to learn about the basics of cell culture research, and if you are going to start the cell culture experimentation. So please come and listen this basic lecture so you can understand the necessities of the cell culture research, that what you need, what are the advantages, the disadvantages of using the different type of thing. There is another slides about the cell culture media. You can see that why we use the cell culture media. That is very important component to create. Previously we were talking about the cell culture environment. So I told you that these all things, these are included to creating the cell culture research environment. So in the media they having very necessary nutrients inside, also the growth factors, the hormones for the cell growth, also the regulating the pH of the culture. They maintain the osmotic pressure of the cell culture. So this was some more about the cell culture media. So this is another slide. In the same still we are talking about the cell culture medium, usually the cell culture environment. So the media having a greater role in producing a very good environment for the cell culture research. So this is the basal media function. The basal media, they maintain the pH and the osmolarity, the required osmolarity usually we required for the cell culture research. Also they provide the nutrients and energy sources. Uh, sometime depend upon the different properties. They depend upon the different compositions of the cell media. Sometime we compose the media according to our own needs. Sometime we have a different experimentation models in which we sometimes we develop our own media for the different type of experiment. Like I have learned, I was reading a cryopreservation study in which they have created their own media using some biological compounds, and they replaced some components of the typically used, you can say, the commercial media. So there is another slide. They are telling you the composition of the basal media. So you can see the composition, the keto acids, then the main carbohydrates, then the vitamins, then the trace elements. So this is the composition of the basic composition of the cell media. So this slide also indicates the functions of these different ingredients in the cell culture media. So this is about the nutrients of the culture media, that the basal media usually we use to maintain the pH and osmolarity, I previously told you, and they different provide the different nutrients and energy sources they are acting as. So the inorganic salts, the primary function is to maintain the osmolarity, to regulate the membrane potentials, especially sodium, potassium and the calcium. These potential are very necessary. These membrane potentials are very necessary usually to grow the cells in the in vitro environment. And then the provide ions for the this for the cell attachment and enzyme cofactors. The pH indicator, phenol red, usually we use this for the optimum cell growth. Usually we indicate this pH, required pH, usually 7.4. And then the buffers, the bicarbonates, you can see the bicarbonate buffered media that required the carbon dioxide atmosphere. So the other things that glucose, usually used for the energy source, the energy required for the cell growth and attachment and the maintaining their multiplying numbers. So this another slide. This same the for the supplements. So L-glutamine, previously I think we discussed, and then the non-essential amino acids. These are very important. They usually added to the basic media compositions, and I previously told you that the composition of these supplements depend upon the nature of the experimentation. Sometime you do certain experiments where you need certain type of media. So these all things depend upon the depend upon the different research projects, the nature of the different research works. And so this dear student, this is was some basic requirements we have studied in this lecture. Whenever we going to start the cell culture experimentation, we have to it is very necessary that we have some know-how about the basic compositions and materials, and especially our main focus was that the cell culture environment. And we have learned that which are the factor responsible for making the good environment for the research. So today's lecture, that is all for the today's lecture. And the next lecture we will learn about some cell culture assays. Also we will learn about the cell toxicity, the cytotoxicity, also about the cell viability. So stay tuned, and for the next lecture. Thank you very much. So nice of you. Take care of yourself. Bye bye and Allah Hafiz. So, viewers, stay tuned and take care of yourself. I will try my best to come with the new lecture same week. So, take care. Bye bye. Allah Hafiz. Thank you.